Welcome to British BioMedicine Institute. Today, I, Dr. Pramod Khatri, Medical Director of British BioMedicine Institute, is going to represent a simple topic on chromosomal anomalies. Chromosomal anomalies are also being known as chromosome abnormalities, chromosomal aberrations or chromosomal mutations or chromosomal disorders which have a missing extra irregular portion of chromosome DNA. If there are chromosomal anomalies, then there are 50% chances of miscarriage in first trimester, whereas there are 5% chances of stillbirth and 0.05% of live bones, which include Down syndrome, Fragile X syndrome, and the chances of somatic cell abnormality in the cancer usually increases. The major cause of congenital malformations can be through teratology. Teratology is a study of birth defect, and a teratogen is something that either induce or amplify abnormal embryonic or fetal development and cause birth defect. Various factors responsible for congenital malformations can be either genetic factors, which can lead to chromosome abnormality, and also there are various environmental factors like drug, thalidomide, steroid, anticonvulsant, alcohol, pollutant, pesticides, estrogens, and various infections like rubella, cytomegalovirus, toxoplasmosis, and maternal illness. The types of chromosomal abnormality can be divided into two types: numerical or structural abnormality. Unifactorial genes can lead to achondroplasia, hemophilia, muscular dystrophy, whereas multifactorial genes can lead to spina bifida, club feet, diabetes mellitus, and schizophrenia. Chromosome abnormality usually occur when there is an error in cell division following meiosis and mitosis. Chromosome abnormality can be detected or confirmed by comparing an individual karyotype or full set of chromosomes to a typical karyotype for the species via genetic testing. During structural chromosome abnormality, aberrations are used to specify structural abnormalities and precede the chromosome involved in aberration of nomenclature string. Deletion, also known as deletion results in loss of chromosome segment where terminal deletions are caused by a single break with loss of segment to a break. There can also be interstitial deletion, which result from two breaks in a chromosome, loss of intervening segment, reunion of the breakpoint. For example, del 5 p15.3. This describes a terminal deletion of short arm of chromosome 5. All chromosome material distal to band p15.3 is missing, whereas del 20 q11.2 q13.3 there is a interstitial deletion of long arm of chromosome 20, and the material between band q11.2 and q13.3 is missing. This is an example of deletion chromosome where a specific portion has been deleted. Ring chromosomes are denoted by R, which are donut-shaped structures that may involve one or more chromosomes. For example, 46XXR7P22Q36. This describes a ring derived from chromosome number 7. A break have occurred at band P22 and Q36, and the end of the segment between the breakpoint have rejoined. The acentric, which is without a centromere segment, distal to the breakpoint have been lost. This is an example of ring chromosome where a chromosome has been bent in a ring shape. When two chromosomes are involved and monocentric one centromere ring chromosome and acentric segment result, their DER should be used, also known as derivative DER or recombinant REC chromosome. 46XY DER 18 P11.2 Q22. This indicates a ring derived from segment between the breakpoint P11.2 and Q22 of chromosome number 18 and acentric fragment of unknown region. If the origin of a ring chromosome is not known, it is listed after all known aberrations but before other markers. For example, 51XX plus 8 plus 13 plus R1 plus R2 plus MAR. This indicates that two distinctly different clonally occurring rings and a marker chromosome are present. If multiple rings are present but it is not known if any of the rings are identical, the rings are denoted by a plus sign and the number of rings identified, for example, the presence of the 3 rings, are described as plus 3R. Next is inversion, also known as INV. In an inversion, a chromosome segment breaks, reorient itself to 180 and reinsert itself. If an inversion involves the centromere with one break in each chromosome arm, it is said to be pericentric. For example, 46XX INV 16 P13.1 Q22. This is a pericentric inversion of chromosome 16. A break has occurred in the short arm at band P16P13.1 and the long arm at band 16Q22. The chromosome segment between these bands have present but inverted. These aberrations are commonly observed in acute myelomonocytic leukemia with eosinophilia. Next example is 46XY inversion 3 Q21 Q26.2. This is a pericentric inversion involving band Q21 and Q26.2 in the long arm of chromosome 3. This rearrangement has also been seen in acute myeloid leukemia. This is an example of inversion chromosome where the material of the chromosome has been inverted. Next is numerical abnormalities. If there is a gain or loss of all chromosome in the karyotype string, then it is usually denoted by the number of either a plus or a minus sign before aberrant chromosome. For example, 47XY plus 21. The exception is sex chromosome in the constitutional studies where sex chromosome gain or losses are indicated by listing the chromosome present. Example, 45X or 47XXY without use of plus or minus sign. Acquired sex chromosome aberrations are written with plus or minus sign. Next is ploidy, which refers to the number of set of chromosomes present. Thus, diploid refers to the normal situation of two sets of each chromosome. Example, 46XX or 46XY. A haploid, triploid or tetraploid karyotype is evident from the chromosome number. For example, 23X, 69XXY or 92XXYY respectively. If additional chromosome changes are evident, these are expressed in relation to the appropriate ploidy level. The ploidy level most commonly used in human karyotyping, most often are in acquired disease, are near haploid 1N, which describe chromosome count up to 34 chromosomes. Numerical abnormalities are expressed in relation to 23 chromosomes. Near diploid 2N, which describe count with 35 to 57 chromosomes. Numerical abnormalities being expressed in relation to 46 chromosomes. Near triploid 3N, which describe karyotypes with 58 to 80 chromosomes. Numerical aberrations are expressed in relation to 69 chromosomes. Near tetraploid 4N, which describe karyotype with 81 to 103 chromosome. Numerical changes are expressed in relation to 92 chromosome. Next is 25X plus 4 plus 10. This represents near haploid karyotype with 2 copies of chromosome 4 and 10 and a single copies of all other chromosomes. 70XXY plus 13. This describes a near triploid karyotype with 4 copies of chromosome number 13 and 3 copies of other chromosome. In numerical chromosomal abnormalities, there are changes in the number of chromosomes which can either be polyploidy or heteroploidy. Polyploidy happens when somatic cells contain multiple of haploid number of chromosomes like 3n, 4n, 5n, etc. Whereas aneuploidy, also known as heteroploidy, are the deviations from the diploid number of chromosomes like 2n plus 1, 2n minus 1, etc. The mechanism of polyploidy includes there is failure of pulling apart of 2 chromatids to opposite end after metaphase stage of the mitosis, reduplication of chromosome without dissolving of nuclear membrane, and there can be failure of cytoplasmic division. Polyploidy can be divided into either autopolyploidy or allopolyploidy. Autopolyploidy is even number multiplied of haploid number of chromosomes, example tetraploidy, 23 into 4 or 92 chromosomes, hexaploidy, 23 multiplied by 6 or 138 chromosomes, or octaploidy, 23 multiplied by 8 or 184 chromosomes, etc. Next is allopolyploidy, odd number multiplied of haploid number of chromosomes, example triploidy, is 23 multiplied by 3 or 69 chromosomes, which is commonest. Pentaploidy is 23 multiplied by 5 or 115 chromosome, and heptaploidy have 23 multiplied by 7 or 161 chromosome, etc. The mechanism of aneuploidy includes if there is a non-disjunction happens when there is a failure of separation of chromosome during cell division, if there is formation of 2 types of gametes, both abnormal, if there is fusion of either of these abnormal gamete with normal gamete can result in trisomy or monosomy. It may also involve autosomes or sex chromosome. Through this diagram, we are explaining non-disjunctions. In general, non-disjunctions can occur in any form of cell division that involve ordered distribution of chromosome material. Higher animals have 3 distinct forms of such cell division. Meiosis 1 and meiosis 2 are specialized forms of cell divisions occurring during generation of gamete, egg and sperm for sexual reproduction. Mitosis is the form of cell division used by all other cells of the body. Meiotic Nondisjunction usually happens due to advanced maternal age. Usually so, meiosis 1 starts at 20-week fetal growth and there will be a rest for 10 to 45 years and finishes meiosis 1 at ovulation and meiosis 2 at fertilization. During trisomy of chromosome, there will be presence of 3 copies of chromosome. Trisomy of autosomes 13, 18, 21 also happens. There are trisomy of sex chromosomes XXX, XXY also. For example, the trisomy of autosome include Patau syndrome, which is trisomy of chromosome number 13. Edwards syndrome have a trisomy of 18 number chromosome, whereas Down syndrome have a trisomy of chromosome number 21. Trisomy of chromosome number 13 is also known as Patau syndrome. It was first described by Barthelion in year 1657 and was redefined by Patau in year 1960. For female chromosomes are 47XX plus 13 and for male chromosomes are 47XY plus 13. If it affects phenotypically both male and female subjects, whereas the incidence rate is 1 in 12,000 subjects, which can increase with the age of the mother. The clinical features of the Patau syndrome includes mental deficiency, low birth weight, abnormal development of frontal lobe, absence of corpus callosum, hypoplasia of cerebellum, sloping forehead, scalp defect, malformed ear, congenital heart defect, renal tract anomaly, microphthalmia, bilateral cleft lip palate, polydactylity with rudimentary digits, and rocker bottom heel. This is a photo of a Patau syndrome, and we can easily see that in the karyotype there is a trisomy of chromosome number 13. Next is the karyotype. Edward syndrome. Next is Edward syndrome happens due to trisomy of chromosome number 18. For female chromosome are 47XX plus 18, and for male chromosomes are 47XY plus 18. It affects phenotypically both male and female subjects, whereas the incidence rate is 1 in 8,000 patients. The clinical feature of Edward syndrome includes maternal deficiency, growth retardation, prominent occiput with elongated head, webbing of the neck, short sternum, micronathia, low set malformed ear, ventricular septal defect, renal anomalies, clenched fist with overlapping of finger, and hypoblastic nails. This is an example of Edward syndrome where various clinical features are visible clearly in the patients. Next is Down syndrome. Down syndrome happens when there is a trisomy of chromosome number 21. For female chromosomes are 47XX plus 21, and for male chromosomes are 47XY plus 21. It affects phenotypically both male and female subjects. The incidence of Down syndrome is 1 in 800, which usually increases with the age of the mother. The clinical features of Down syndromes are short height, malformed large ear, severe mental deficiency with decline in IQ with age, epicanthal fold of the eye, bruise-filled spot in iris, brachycephaly with flat face and occiput, renal anomalies, prominent or protruding tongue, scrotal tongue, flat and low nasal bridge, upward slant or palpebral fissure, CMN crease, and clinodactylity of the fifth digit. This is the clinical features which I have already discussed for the Down syndromes. They are again visible, clearly visible in the Down syndromes. Next is microdeletion syndrome. For microdeletion syndrome, the most common example is WBS, Williams-Beuren syndrome, where the incidence rate is 1 in 20,000 of all the populations. Phenotype characteristics include dysmorphic faces, growth and mental retardation, distinctive personality, transient hypercalcemia, arterial disease, uniform 1.5 MB deletion del 7q 11.23, region flanked by duplicated gene, non-homologous recombination, 17 gene including ELN which encode tropoelastin point mutation caused AD supravalvular aortic stenosis. This is an example of a Williams syndrome, and various clinical features are clearly visible. Next is Prader-Willi syndrome, also known as PWS. The incident rate is 1 in 10,000 population. The phenotypic characteristics include mild to moderate MR, hypertonia, poor feeding in infancy, short stature, small hand and feet, small external genitalia, hyperphagia, compulsive overeating, and obesity. There has been deletion of paternal gene which is del 15 Q11 to 13. There can also be due to uniparental disomy in some cases. This is an example of a PWS patient, and various clinical features are visible in this patient. Next is Angelman syndrome, also known as AS. Patient has severe mental retardation and there are absence of speech. There are jerky movement with inappropriate laughter. Patient usually have large jaw. Angelman syndrome happens when there is deletion of maternal gene which is del 15 Q11 to 13. In this, we are comparing both Prader-Willi syndrome and Angelman syndrome. Prader-Willi syndrome, light pink color is represented by mother, whereas light black color is presented by the father, where it is concluded that 70% cases father is responsible for Prader-Willi syndrome, and whereas in Angelman syndrome, light pink color is represented by mother, whereas light black color is presented by the father, and it can be concluded that 70% cases mother is responsible for the syndrome. Next is genomic imprinting. Is an epigenetic phenomenon that causes gene to be expressed in a parent of origin specific manner. Gene, however, can also be partially imprinted. Here, maternal and paternal genetic contributions are not equivalent, whereas genetic contribution from both the parents are needed for normal development of the embryo. Evidence of imprinting can be found in mouse embryo, where gynogenetic poorly developed extra-embryonic membrane and in androgenetic abnormal embryonic structure is present. Genomic imprinting can also lead to human tumor, whereas hydatiform moles, placental tumor can occur with two paternal haploids, set of chromosomes, and ovarian teratoma can develop if there is a benign differentiated tumor with two maternal haploid set. The mechanism of imprinting: in some cases, genes are preferentially inactivated or are imprinted during gametogenesis in male and female parents. There can be differentiated DNA methylation, histone acetylation. There can also be deletion of active allele such as functional nullisomy, or due to uniparental disomy for the inactive allele like functional nullisomy. Here, I am presenting few MCQs in order to check your understanding of the topic being taught. Please try to provide the correct answer in the comment section of YouTube. The first MCQ is: Patau syndrome is associated with trisomy of chromosome 13, 15, 18, and 21. Next MCQ is: Rocker bottom heel is associated with Patau syndrome, Edwards syndrome, Down syndrome, or none of the above. Third question: Bruce-Field spot is a characteristic feature of Patau syndrome, Edwards syndrome, Down syndrome, none of the above. Fourth question: all of the following are trisomy of autosomes except Edwards syndrome, Klinefelter syndrome, Down syndrome, Patau syndrome. Fifth, all of the following are example of polyploidy except triploidy, tetraploidy, hexaploidy, and octoploidy. Please provide your answer in the comment section of the YouTube box. Thank you for showing interest in British Biomedicine Institute Scale Postgraduate Nano Degree Program in Genetic and Genomic Counseling. Hope you have gained a lot of clinical information through this presentation. Please share, like, and subscribe British Biomedicine Institute channel. Good luck.